Hello everyone, welcome to my channel Biochemistry Concepts. This video is about components of electron transport chain. We know that there are four complexes through which electrons flow in the electron transport chain. These complexes are made up of different components. To understand what are components in a simple way, let us consider one example, that is mobile. The different parts of the mobile are components of the mobile, like it has circuit, it has antenna, it has display, flash, processor, speaker, volume buttons. So all these are the different parts of the mobile. We call these as the components of the mobile. In the same way, each complex of the electron transport chain is made up of different parts. So these are called as the components of the electron transport chain. Electron transport chain contains 5 types of components. Those are nicotinamide nucleotides, flavoproteins, iron-sulphur centers, ubiquinone and cytochromes. These are responsible for the transfer of electrons in the ETC. Now we'll discuss in detail about each component. Coming to the first component, nicotinamide nucleotides. The water-soluble vitamin, that is niacin or vitamin B3, it has two active forms or two coenzyme forms. Those are NAD and NADP. So NAD is nicotinamide adenine dinucleotide. This NAD is more actively involved in the electron transport chain. NAD plus is the oxidase form, whereas NADH is the reduced form. So this NAD plus is converted to NADH by dehydrogenases by transferring a pair of electrons from the substrate. So when a pair of electrons is transferred to NAD plus from the substrate, NAD plus is converted into NADH, and these reactions are catalyzed by dehydrogenases. NADH transfers its electrons to the initial component of the electron transport chain, that is NADH dehydrogenase. It is a large protein complex. Coming to the other active form of niacin, that is NADP, and its full form is nicotinamide adenine dinucleotide phosphate. The reduced form of this NADP, that is NADPH, it is involved in the biosynthetic reactions. For example, in the synthesis of fatty acids and cholesterol synthesis, NADPH, that is the reduced form of NADP, is required. Coming to the next component, that is flavoproteins. Proteins with FMN or FAD as prosthetic group are called as the flavoproteins. These flavoproteins are important hydrogen carriers. They are important components of complex 1 and complex 2. Coming to examples of the flavoproteins, NADH dehydrogenase, that is complex 1 of electron transport chain, is a flavoprotein because it contains FMN as the prosthetic group. So in the complex 1, when NADH transfers electrons to the complex 1, FMN is reduced to FMNH2 and NADH is converted into NAD+. So in this reaction, FMN accepts 2 electrons and 2 protons and it is converted into FMNH2. Same way, succinate dehydrogenase, which is found in the inner mitochondrial membrane, is also a flavoprotein because it contains FAD as the prosthetic group. So here when you see the reaction, succinate plus FAD, they react to form fumarate and FADH2. So here the electrons from the succinate, they are transferred to FAD. Same way as FMN accepts the electrons and protons, here also FAD accepts 2 protons and 2 electrons and it is reduced to FADH2. Coming to next component, that is iron-sulphur proteins or iron-sulphur centers or non-heme iron proteins or iron-sulphur clusters. So these are found in complexes 1, 2 and 3. Here iron is not present in heme, but it is associated with sulfur atoms of cysteine residues in the proteins or with the inorganic sulfur atoms or both. So these iron sulfur centers may exist as simple structures with a single iron atom coordinated to 4 cysteine sulfhydryl groups or more complex iron sulfur centers with 2 or 4 iron atoms. The iron sulfur takes part in the single electron transfer reactions in which one iron atom undergoes oxidation reduction between Fe2 plus and Fe3 plus form, that is ferrous and ferric states. Coming to next component, ubiquinone or coenzyme Q. It is derived from the word that is ubiquitous, which means existing everywhere. So this ubiquinone is a benzoquinone with hydrocarbon tail. Because of this, it is strongly hydrophobic, and that's why it is confined to lipid bilayer of inner mitochondrial membrane. It is a mobile lipophilic electron carrier. So it can accept electrons from the complex 1 or complex 2 and transfers them to the complex 3. So coenzyme Q also links flavoproteins to the cytochromes. That means complex 1 and complex 2 are flavoproteins. These are linked to the complex 3, which is a cytochrome. So that's why you can say that coenzyme Q links the flavoproteins to the cytochromes. So coenzyme Q, when it accepts one electron and one proton, it is converted into semiquinone. And this semiquinone, again, when it accepts one more electron and proton, it is converted into ubiquinol. So coenzyme Q is oxidase form and coenzyme QH2, that is ubiquinol, is reduced form. Coming to next component, that is cytochromes. These are the final class of components that participate in the electron transport. So these are integral membrane proteins except cytochrome C, and these are named as cytochromes because these are the colored cellular components. Here cyto means cell and chrome means color, and these are conjugated proteins containing heme as the prosthetic group. So heme contains iron plus porphyrin ring. Heme group of cytochromes is different from the heme group of hemoglobin and myoglobin. The iron of heme in cytochromes is alternately oxidized and reduced. That means it changes from Fe3 plus form to Fe2 plus form. So there is always interconversion between these two forms, which is essential for the transport of electrons in the electron transport chain. But iron in heme of hemoglobin and myoglobin remains in the ferrous state only. The cytochromes were designated as A, B and C depending on the type of heme present and respective absorption spectrum. Electrons are transported from coenzyme Q to cytochromes in the following order: B, C1, C, A and A3. The property of reversible oxidation reduction of heme iron, that is interconversion between Fe2 plus form and Fe3 plus form of cytochromes, allows them to function as effective carriers of electrons in the electron transport chain. Cytochrome C, it is a small protein with 104 amino acids and a heme group. It is water soluble protein and it collects the electrons from the complex 3 and delivers them to the complex 4. Cytochrome A and A3, also called as the cytochrome oxidase, is the only electron carrier, the heme iron of which can directly react with the molecular oxygen. Besides heme, this oxidase also contains copper that undergoes oxidation reduction, that is between Cu2 plus and Cu plus during the transport of electrons.